Hello and welcome to SRG's Medicine. Today, in this video of Respiratory Medicine Series, we shall be talking about abnormal breathing patterns. So, the contents of this video are finding respiratory nuclei, present view on respiratory nuclei, fun facts, and ultimately summary. So, before starting the quest on finding the respiratory nuclei, I'd like to say that scientists, for determining the exact location or roundabout location of the respiratory nuclei used to cut the brain and spinal cord at different levels and observed the changes in breathing patterns. So the first name that is to be mentioned here is Galen, who was there in 2nd century as a physician for the gladiators in the Greek city of Pergamon. Now, as a physician for gladiators, he used to see a lot of violent battles, and the patients used to be injured gladiators on battles, and it was quite common for them to have their swords driven right through the neck and taken out from the back of the neck. So it could lead to injury in the spinal cord or brainstem as well. So he found out that breathing stopped after a sword blow, which was given to a higher cervical spine. But similar blow, which was given to a lower cervical spine, left the arms as well as the legs paralyzed. However, the respiration continued. Now, he thought, like as a physician and also with the scientist mind, that let's see if that's true even in live animals. So he thought about reproducing these lesions in live animals, and after many experiments, he ultimately concluded that yes, brain truly sends the information via the mid-cervical segment of the spinal cord to the diaphragm. So the respiratory controlling nuclei must be somewhere in the mid-cervical spinal cord. Well, he was not right, but he tried his best according to the time in which he lived. So after a long time, that is around 18 centuries later, Thomas Lumsden in 1923 AD, who was working on the Department of Experimental Pathology in Lister Institute of Preventive Medicine, which is a quite popular building in the UK. And here he worked on cats. Yeah, well, cats, because I don't know what scientists have the fascination with cats. You can take the example of Schrodinger, who used to imagine keeping cat in a box, sending it in space, killing it with the poison, and thinking whether it would be dead or not. So similarly here, but here he performed real experiment, not just imagination. So here, what he did was he transected the spinal cord and brainstem of cat at different levels and observed the changes in respiration to find the location of respiratory nucleus. Now here I have a picture from the original article, which he published in 1923 AD. Now this picture depicts the brainstem along with the spinal cord and some part of spinal cord and also the brain of the cat. And here there are different lines kept there named 1, 2, 3, 4, 5, and 6. Now these are for the incisions where he gave, or the area in which he gave incisions. So let me classify you the location of specific locations here before starting. The first one, which is in blue, that is the upper pons. Now the one in the red, that is the lower pons area, and also some parts of the pontomedullary junction. Whereas the lower one in green, now that is medulla oblongata, and below that there's a spinal cord. So now here we can see that after transecting between number 5 and 6, between the line number 5 and 6, so that is around this area, he found out that the ventilation ceased. However, that was due to the loss of input to funicular as well as intercostal nerves. But the muscles on whose the innervating cell bodies lies in the upper parts of the brainstem still work. For example, flaring of nostrils, tongue, pharynx, larynx, and that also in a periodic pattern, as you would see in a normal respiration. So he concluded that a spinomedullary transaction, so that is in the specific location between the spinal cord and medulla oblongata near the spinomedullary junction. Here, a spinomedullary transaction will block the ventilation by interrupting the output to diaphragm, but not by interrupting the respiratory rhythm. Hence, the central pattern generator for the respiratory rhythm lies somewhere above the spinal cord in the brainstem, not in the spinal cord. So he disapproved what Galen had said about 18 centuries ago. So after that, here cutting this location on 5 and 6, he noted some gasping. So he noted it as gasping nucleus. And between number 4 and 5, respiration ceased after cutting number 4 and 5, and here he noted it to be expiratory nuclei. And on number 3 and 4, so when cutting between number 3 and 4, he noted that there was presence of inspiratory apneusis after cutting the location of the lower horns. So that is between the line 3 and 4 in this red line. Here, what we mean by apneusis is that there is deep inspiration followed by a sudden brief expiratory pause, and again there's deep inspiration. Like, yeah, so that kind of inspiratory pattern is called as inspiratory apneusis, and he noted it to be due to some kind of nuclei located there, which he named subsequently as apneustic center. Now, after that, the incision between the line 1 and 2 gave rise to another nuclei, which he identified as pneumotaxic nucleus. Now, that he thought was there to counteract the apnea. So that was what he found. Now, let's move on to our next individual, that is Robert Gessel and his colleagues. So here you can see a young man there in 1930s. Now, he, along with his colleagues, used the help of extracellular microelectrodes in the spinal cord and also in the extracerebral fluid, or in the CSF. Here, he noted that there are many neurons which are present in medulla which will increase their firing rate only during a specific time, whether during inspiration or expiration. So only during a specific time of respiration, not during the both. So here now he classifies the respiratory nuclei as inspiratory or expiratory. And he concluded some important findings. The first one, that not all neurons which fired in phase with respiratory cycle, which we previously in the first video described as respiratory related neurons, or RRN. So not all RRN are involved in control of breathing. Here you can take an example of aortic baroreceptors, because these baroreceptors produce an output which varies in phase with the lung inflation and the ventilation, but they are primarily involved in cardiovascular functions or maintenance of the cardiovascular function, not respiration. So not all RRN are involved in control of breathing. And the next thing he found out was that there are some neurons which do not fire relating with the respiratory cycle or in phase with the respiratory cycle, but they may be essential for respiratory control. Now here you can take an example of central chemoreceptors, because they fire tonically and not in phase with the respiratory cycle, but they are quite essential, as we know, for respiratory drive and their maintenance. So ultimately, after all of these findings, he concluded that not all respiratory related neurons play a direct role in respiration. However, respiratory control involves more than just RRN. So that was his finding. So that's all about our quest on finding respiratory nuclei. Now we'll be talking about our present view on respiratory nuclei. So here's a picture for that. And this shows that there are presence of two locations, specifically two places for respiratory nuclei. The first one being in the medulla, second one in the bones. So the medullary nuclei can further be classified into dorsal group and ventral group. whereby the ventral group lies in the ventrolateral section, whereas dorsal lies near the base of the fourth ventricle, near nucleus tactus solitarius. So that's about the medullary group of respiratory nuclei. And about the pontine group, there are two nuclei, namely pneumotaxic center and apneustic center, and their roles will be classified later on. So for now, we're just looking about the locations of these respiratory nuclei. So after that, here is a table that will give you an idea about the medullary respiratory nuclei, and we'll be discussing it on the basis of location, major component, and also on the dominant activity. So the first one, that is dorsal respiratory group. It's located, as the name suggests, in the dorsal part of the medulla, and the major components here include the some parts of nucleus tactus solitarius and the neurons which lie around the nucleus tactus solitarius. So these are the major components. And I'd like to make you clear here that nucleus tactus solitarius is actually what we know as a sensory integration center for autonomic functions. And because they contain the termination of both vagus as well as glossopharyngeal nerve, and they transmit the signal into the respiratory centers from three things. The first one, peripheral chemoreceptors; second one, baroreceptors; and the third one, other different types of receptors which are located in the pulmonary parenchyma. So in the first part of the videos on this series, we also mentioned that there's some sensory integration center, and now what we see here is that nucleus tactus solitarius is that same sensory integration center. Now after that, its main or dominant activity is in inspiration, and specifically dorsal respiratory group functions during quiet respiration because it functions for quiet inspiration, and normal quiet expiration is just an elastic phenomenon because of lack of the inspiratory activity. So now we'll be talking about the ventral respiratory group. And the ventral respiratory group can further be divided into the rostral part or upper part, intermediate part, and the caudal or lower part. And all of their location is in the ventrolateral part of medulla. And rostral part includes nucleus retrofacialis, or also quite famously known as Botzinger's complex. Now, the story behind why the Botzinger's name was kept, that's quite an interesting topic, and we leave it for the upcoming positive videos. And for now, let us just focus that here, its dominant activity is during expiration. But the twist here is that it doesn't directly cause expiration. Rather, it only acts as an interneuron which connect the dorsal respiratory group with the caudal part of respiratory group. So that is just acting as an interneuron for the expiratory signals to be sent to the caudal part. And here we have to note that whenever the drive from the dorsal respiratory group becomes dominant or excessively high, then in such cases there are spilling of some information or spilling of the stimulus, and that impulse which spills over is collected by the rostral respiratory group, and that is taken to the caudal part, helping in expiration during a forced condition or forced expiration, and also captured by intermediate one for forced inspiration. So now we'll talk about the intermediate one, which includes pre-Botzinger complex, nucleus ambiguous, and nucleus parambigualis. Now here, nucleus ambiguous and nucleus parambigualis will act as a somatic motor neurons, which move via cranial nerve number 9 and cranial nerve number 10 to different parts of the respiratory system, thereby helping to increase the caliber or diameter of the upper airways during inspiration. So that's the major role of the intermediate group, and the role of pre-Botzinger's complex is nowadays thought to be acting as a central pattern generator. So what we knew as CPG, or the pacemaker of the respiratory system, is actually pre-Botzinger's complex, which is located in the intermediate part of the ventral respiratory group. That is located on the ventrolateral part of medulla oblongata. So talking about caudal group, which includes nucleus retroambigualis, and it is involved in the expiration because it acts as a expiratory premotor neurons. So here, the thing to be noted is that it is not a motor neuron but acts as a premotor neuron, which travels via the spinal cord and then synapses on the motor neurons of accessory expiratory muscles, which includes the muscles of the abdominal wall, muscles of the intercostal area or intercostal muscles, sternocleidomastoid muscles, muscles of the back, and other expiratory muscles. So here, taking this whole table at a look, we can take three conclusions. The first one, dorsal respiratory group is involved in quiet respiration, whereas ventral respiratory group is involved in forced inspiration as well as expiration. Here, dorsal was involved specifically for inspiration, whereas ventral both for inspiration as well as expiration. The next thing is that the pacemaker for respiration, or also known as CPG, is located in the pre-Potzinger's complex in the intermediate VRG. So that was about what we need to conclude on this table. So we'll be now talking on the mid-pontine centers, which include apneustic and pneumotaxic center. So previously, apneustic center was believed to cause apnea, and we all know what was believed to cause apneusis. We all know what apneusis is. And we'll also be talking about apneusis in detail in the upcoming video, but for now, just that much. And then pneumotaxic center was thought to counteract apneusis. But now, here's a twist. Nowadays, based on the electrophysiological studies, we believe that apneustic center is a non-existent center. So the existence of apneustic center itself is put into doubt because there's no proof that we have gained for the existence of the apneustic center via electrophysiological studies. And the next thing is that pneumotaxic center nowadays is thought to control respiratory rate rather than preventing the apneusis. Because the proof for that is that the strong pneumotaxic signal can increase the respiratory rate to 30 to 40 breaths per minute, whereas a weak pneumotaxic signal can decrease the respiratory rate to 3 to 5 breaths per minute. So the rate is quite dependent upon the pneumotaxic signals, as we can see here. So that's our present view on the respiratory nucleus. Now we'll be moving on to some fun facts. So fact number one, a change in blood carbon dioxide concentration has a potent acute effect for controlling respiratory drive, but only a weak chronic effect after a few days of adaptation. So the adaptation usually takes 1 to 2 days, and after 1 to 2 days, the constant increase in carbon dioxide level will not cause much rise in the ventilation. Now, this is believed to be due to renal factors or renal compensation, because after 1 to 2 days, renal compensation takes place, due to which hydrogen ion produced by excessive carbon dioxide are just thrown away and bicarbonate is retained. So bicarbonate will combine with the carbonic hydrogen ion formed from the carbonic acid, ultimately preventing the diffusion of the dissociation of carbonic acid into H plus and bicarbonate ion. So thereby it will decrease the acidity, so that cannot stimulate the acidity also in the CSF, so that will not be able to stimulate the central chemoreceptors. As we had talked also in the previous video, how CO2 causes changes in the central chemoreceptors, thereby causing it to be stimulated. So the next fun fact here is that CO2 is not the major controller for respiration, but oxygen is not the major controller. And why is that? Because during normal conditions, we do not see much variation of oxygen level. That's specifically due to hemoglobin oxygen buffer system, or as we know in the sigmoid-shaped hemoglobin dissociation curve. And here, the thing is that only under severe condition does the blood oxygen level decrease to about 30 to 60 millimeters of mercury. And that range is required for activation of the peripheral chemoreceptors. So it wouldn't be quite useful for a normal day-to-day control of ventilation or minute-to-minute control of ventilation. But in the case of CO2, even the slightest change in ventilation would cause a large or massive change in the arterial CO2 concentration. So that can be a quite good indicator for ventilation. Hence, CO2 acts as a major controller for respiration, but not oxygen. And the next thing is that neurogenic control of ventilation during exercise is partly a learned response. Now, here I'd like to give you an example that if you're just beginning on gym, then after a few push-ups, probably about 10 to 20, if you're good enough, then after that you'll be there gasping for air with both your nose and mouth open and roaming here and there for air. But the same thing. So a trained person who works regularly on the gym can do about 50 to 100 push-ups easily, breathing only by nose, all the while respiratory rate remaining the same. So ventilation doesn't change to him much, but just the thing is that he does it quite effectively as compared to you because he has learned it due to repetitive practice. And we know that a learned response requires repetition. So that's what we say, that is neurogenic control of ventilation during exercise is a partly learned response. And the fourth thing is that peripheral chemoreceptors fire tonically and they will increase their firing rate during hypoxia, specifically in the range of 30 to 60 millimeters of mercury. Now here you can see that in this picture. In the x-axis is arterial PO2, partial pressure of oxygen, in mmHg, whereas in the y-axis there is carotid body stimulus or impulses per second. Now here you can see that during the normal range, so within 200 mmHg at max, there you can see that there is the impulses per second of carotid nerve firing is not zero. It's some positive number. So there's always tonically firing. And this is like quite unrealistic or unachievable condition. So this is just extrapolation up to 500 mmHg. And this is not seen, like zero is not seen in a normal situation. So the peripheral chemoreceptors fire tonically and on the conditions of hypoxia, specifically from 30 to 60 mmHg. Now they increase their firing rate rapidly. Now, that is what I mentioned in the fourth point. So these are all with the fun facts. Now summarizing this video, there are four types of respiratory nucleus named as dorsal respiratory group, ventral respiratory group, apneustic center, and pneumotaxic center, whereby dorsal respiratory group acts for the normal quiet respiration, specifically only for inspiration, and expiration is an elastic process which occurs by itself spontaneously after cessation of the inspiratory stimulus. And ventral respiratory group acts for forced respiration, both inspiratory as well as expiratory, and not to forget that there's also presence of pre-Botzinger's complex, which acts as a central pattern generator or pacemaker of the respiratory system. And apneustic center is thought or was thought to be involved in apnea, but its identity itself is now at stakes. And pneumotaxic center controls the respiratory rate and also was thought to prevent apnea. So that's the summaries of today's video. Hope you enjoyed it. Please stay tuned and do subscribe for getting notifications on the upcoming videos. Thank you. Hope you enjoyed it.